Thank you, everyone, for attending this town hall to tell you about the USC-Buck Nathan Schock Center of Excellence, some of the cores that we have and the opportunities that we have for you to utilize the resources of our Nathan Schock Center to advance your geroscience research. I'm Sean Curran. I'm at the University of Southern California. My counterpart up at the Buck is Gordon Lithgow, and we run the operational side of the Nathan Schock Center. And so today we're going to go over what opportunities are, tell you a little bit about each one of the cores that we have, and then show you our website and how you can apply for funding, talk about some of the different opportunities that are available. And then we've saved time for question and answers at the end for Gordon and myself or any of the core leaders that are here. Everything that we're going to tell you today can be found at our website, USC-Buck-NSC.org, including this presentation, which you can watch later, or you can refer to some of your colleagues if they weren't able to attend. So that. So USC is one of the newer Nathan Schock Centers that was just awarded this past year. It's a true collaboration between the University of Southern California, the Leonard Davis School of Gerontology, and the Buck Institute of Aging Research up in Novato. And so we join a collection of outstanding institutions across the United States whose sole purpose is to provide resources, access to technology, and assistance in your geroscience research. Everything is funded through the National Institutes of Health, through the NIA. So you can apply for access to the resources that we have available. We have opportunities, if you don't know what resources you need to use, we have folks that can help you determine ways that we can assist you in your research. And all of these cores or all of these centers are actually interconnected as well. So if there's something that we can't do, we're happy to refer you to someone else at one of these other organizations that might be able to help you with your research. So the Nathan Schock Centers are named after Nathan Schock. He's sort of thought of as the father of gerontology. He worked at the NIH for almost 35 years and then became a scientist emeritus at the center. He's probably most well associated with the idea of actually needing to use longitudinal methods to really understand aging, rather than looking at individual cohorts of individual ages and not following single individuals or single populations. And so he sort of brought up the idea that the rate of aging of an individual or a group is something that is different and that changes with age. And so he's the perfect person to name after all of these centers as sort of the founder of a lot of this work. So Nathan Schock Centers, I gave you a sort of a brief overview. The idea is to provide leadership to the greater scientific community, and not necessarily specific to the geroscience community, but to bring individuals who maybe aren't working on aging research into the aging field. And so part of our mission is to help you figure out how you can do that, how you can design a study based in your model system or in your specific area of science, and how you might apply that to a geroscience concept. We do that through research development cores. So they are the grant funding component of each of our centers. So you'll apply to the research development core, tell them what you want to do, which core facilities or which services you want to utilize, and they are the ones that provide the support. We also organize a national meeting in order to highlight specific areas of research. And then one thing that's unique to the USC-Buck Nathan Schock Center, we have this idea of mini sabbaticals that you can take. So part of our mission is to train scientists on how to use these technologies and use these resources. And now that COVID restrictions are being lifted, we think this is a really great opportunity for you to come to the Buck Institute or to USC, work one-on-one with one of the experts in geroscience, and learn hands-on how to do these technologies, or work in that lab for a couple of weeks, and we can provide resources and funding to help you do that. We're mostly going to focus on pilot projects and vouchers today, which will be explained in a little bit. But just so you know, now that COVID restrictions are being lifted, that aspect of our Nathan Schock Center is going to come back again. Okay, so the USC-Buck Nathan Schock Center is greater than the sum of its parts. So we have a long standing relationship together, the Buck Institute and the University of Southern California. Most recently, the last 6 years, we've had a biology of aging PhD program where we train PhD level scientists, both at the Buck Institute and at USC. And so that really laid the groundwork for the connections that we have between our two institutions. And I think really made for a more powerful Nathan Schock Center, just both in scope, the available resources that are there, and the scientific community that we built between our two institutions. Just as sort of an organizational structure of how our Nathan Schock Center works. Gordon and I run the administrative component of the Nathan Schock Center in collaboration with our external advisory board and our internal advisory board. We sort of set the direction of what sort of pilots and vouchers we want to fund, whether or not we're staying true to our mission of promoting geroscience research, and most importantly, to make sure that we are providing adequate resources to external organizations. So the majority of the money that we have to provide core services and technology access is for researchers not at USC and not at the Buck. So please apply to them because we have money to give to you. The Research Development Corps is the one that oversees the funding process. And then you will utilize the resource cores, which is the Genomic Translation Across Species Core, the Cellular Senescence and Beyond Core, and the Geroscience Technology Core. And you'll hear about each one of those today. So really, our Nathan Schock Center allows you to engage with geroscience experts and across pretty much every axis of geroscience research. We really are interested in finding new and innovative ways to collaborate with you. We hope to use the Nathan Schock Center to foster those collaborations, provide access to geroscience technology that you may not have at your institution. That's probably the most common voucher request that we have, is to use one of the technologies that we have that isn't available to you at your institution. Train the next generation of geroscience researchers. That's this mini sabbatical idea. This can be junior faculty, postdocs. It can be, even if you're not in the geroscience community at the moment, if you are new to the aging research field, you qualify for both these mini sabbaticals as well as access to pilot and voucher funding. And again, then to provide project support for your investigations. Again, everything is on this website, but I wanted to point out a few individuals who really make our program work. Tara Riley, Linda Hall, and Jillian Miller are in charge of the programmatic aspects of the Nathan Schock Center at the Buck and at USC. All of the financial components, so making sure that the budgets are appropriate and adequate for the types of work that you want to do, are done through our financial partners, Kira Harvath and Sarah Lyons. And then communication, our communications team that monitors both the website and is responsible for helping us do this webinar today, Orly Dolman and Robin Snyder. And again, everything is at the USC-Buck-NSC.org website. Okay, so I'm going to turn it over for a few slides to Simon Mellov, who's going to tell you about the Geroscience Technology Core. Each of our resource cores have a faculty representative from both USC and the Buck, with the exception of two of our cores that are housed solely at each of the institutions. But Walter and Simon, they have actually the hardest job of our team at the Nathan Schock Center, because they are the ones that have to tell you about all of the technologies that are available both at USC and the Buck that are available to you. Simon's going to tell you about a few of them today, and then I'm going to give you a little bit of a really quick version of all of the other stuff that's available. So I will hand it off to Simon, and I'll go through the slides for you. whenever you are ready.
Okay, ready. So, okay, thanks, Sean. So really this core, as Sean said, is to kind of provide advice, although we can do some experiments as well, or facilitate access to investigators who have this technology. You know, if you're successful in getting a pilot award, then those experiments could be carried out. And the goal of the core is to sort of serve as an entry port for each of these different technologies. And it happens at both sites, which makes it kind of complicated, but also interesting. We generally want to promote the use of these unique approaches for investigating different aspects of geroscience. Next slide. So the platforms which we're you know, trying to facilitate access to have a series of horrible acronyms, and they're called the Mitochondrial Metabolism Technology Platform, the MMTP. Remember that one. The second one is an even better acronym, which is the Model Organisms Health Span and Resilience Technology Platform, the MOHRTP, because that just rolls off the tongue. Then the third one is Next Generation Technology Platforms, which encapsulates a range of different, basically omics approaches. And I want to highlight a recent funded example. We've already had one round of pilots funded through the USC-Buck Nathan Schock Center. And one of these was for looking at vascular function in aging, generating data from some of the more, I guess, cutting edge and single cell profiling approaches, which are becoming increasingly popular. I think it's generally accepted that there's a sea change underway in terms of technology applied to biological questions in general, and it's sort of being spearheaded by single cell techniques in a general sense. I mean, they're basically becoming the new PCR. If you're not doing these techniques as a routine part of your investigations, then you're going to be behind the curve. And they've got enormous power to uncover new insights into biology, particularly with regards to geroscience. And so Doug Seals and Zach Clayton at the University of Colorado in Boulder proposed a pilot to investigate what happens in gene expression in the aging vasculature. Now, the aging vasculature is an important problem. There's no treatments for basically the stiffening of blood vessels as we get older. It actually has a huge healthcare burden associated with it. Doug and Zach proposed an experiment to look at the aging aorta in the mouse to better understand what's happening within the actual tissue at the single cell level. And so what they wanted to do was use spatial transcriptomic proteomic profiling. Now, this relies on an instrument which we have at the Buck Institute called the Geomic Spatial Profiler by NanoString. This is a schematic of how the technology works. Essentially, it allows you to generate high-resolution, full genome transcriptional profiles within the context of tissue architecture. You can do this at, as I said, gene expression levels or protein levels, using potentially hundreds of antibodies simultaneously. It's got enormous power. It's also enormously expensive, so bear that in mind. Don't propose to do like 50 samples, otherwise you won't be eating for a year. I'm not going to go through the technology. This is well described, but the experiment which Doug and Zach proposed was to take an aorta from a mouse, young versus an old mouse, or in some cases they might use an accelerated aging model, and basically excise it, section it into 1 millimeter pieces, and then embed some of these in paraffin. We can then section those and profile at the whole genome level to understand what's going on with regards to increasing vascular stiffness with age. And this is a perfect example of a cutting edge technique being developed by the core, because it provides access to instrumentation and expertise, which not everyone has access to at the moment, although I suspect most of you will be using this technology at some point in the next few years, if you're not already, just because of the power of the approach. But this illustrates, again, a potential project utilizing the core services, and it's a nice introduction to the GTC core. So that's all I have to say.
Thanks, Simon. So that was just one example, and you can imagine if we went through every technology, this would be a much longer than a 30-minute or 30 to 40-minute webinar. There's a number of cores that are available both at USC and the Buck for mouse phenotyping, for developing biomarkers, for doing metabolomics, for AI and big data, proteomics, et cetera. And you can find links to all of that on the website. So I'll remind you of that. Again, also, this is also funding for faculty partnerships. So if there is a specific technology in one of the labs at the Buck or at USC that you would like to utilize, take a look at the websites for our faculty at both institutions. Many of our faculty are experts on doing single-cell RNA-seq, on doing small genome sequencing, utilizing a model system. If you are currently doing cell and tissue culture and you want to try something in a whole animal study, you can partner with one of our investigators that work in yeast, worms, flies, the killifish model, or in mouse, and propose studies for that. Again, anything that's being done at USC or the Buck with relationship to geroscience research is a potential collaboration that you can set up. We're happy to work with anyone. And then so that's all I have. I'm going to show you the website really quickly since we've talked about it, and some of our other core leaders in their presentations will show you, or we'll refer to that as well. So let me show you really quickly. So this is the website, USC-Buck-NSC.org. I'm not going to go through all of the links that are up here. The most important one for you is submit a grant proposal, which is this blue link here, or you can go up to grants up at the top. This page will lead you to the portal for submitting a proposal. The basic information is just listed here, and you upload a single file into this box. The requirements are listed at the bottom of this page. So letters of intent for the next round of funding are going to be due on June 30th. It's really simple, a brief, and we mean brief, so less than a page. What is the description of your project? What core do you want to use, and how is that going to accelerate your aging research? And where are you in your career and field? And also an estimated budget. And we understand that you might not know exactly how much that is, but it's a ballpark number at this point. You are encouraged very strongly to have a conversation with the core leaders in order to find out whether or not your project idea is even feasible, and what sort of the scope of the project will be and how much it'll cost. From those letters of intent, we'll ask a certain number to submit a full application. These two are not meant to be extensive exercises for you to get funding. We want a simple face page, just one page maximum, that has the title, who the PIs are, who the other personnel are, and what the abstract is. Your approach page is one page. Really, these are not NIH R01 applications, right? This is one small aspect of a project that's supposed to spark innovation and lead to something that you can use for preliminary data for one of those R01 applications in the future. Biosketches, references, and then a budget justification, and links to those forms are here as well. And again, everything can be submitted into this portal. If you need more resources, you can find them on this page, including a link to all of the presentations that we're showing you here today. This was our last town hall back in February. A recording for this one will be presented here as well. Okay, and with that, I am done. I will turn it back over to our next presenter, which I think is going to be Kelvin Davies from the Research Development Corps.
Okay, trying to get this to work here.
Oh, really quickly, while Kelvin's setting up. So there was one question that was posted in the Q&A. And again, you're encouraged to please use the chat or the Q&A section to ask your questions. So the question is, can undergraduates be granted access to these resources? They can. You're going to need a faculty sponsor in order to submit a proposal or to set up a collaboration. But there's also training opportunities as well. So if you have a very specific question in mind or a resource that you need access to, please send myself or Gordon Lithgow an email, and we can get you in touch with the right person.
Okay, for some reason mine is not working well. Natalie, can you do it for me?
We can see your slide, Kelvin.
You can, I can't. That makes it difficult. You see it now? No, yes.
Yeah, it's up. I don't know if it was.
Okay.
If
You got it now?
Yeah, it's good.
You okay now?
Yes.
Yeah, we can see it, Kelvin.
Sorry about that. So Research Development Core, Julie Anderson and I are directors of that core, provides access to the USC-Buck Nathan Schuck Center resource cores in support of new research initiatives. Provides focus on junior faculty and investigators new to the aging field. That's our primary focus, especially from institutions that may lack aging programs. And would like to move forward. So we basically employ three primary mechanisms to achieve that goal. First is providing support for pilot projects, and Sean was talking quite a bit about that, as was Simon, to address proof of concept and feasibility analyses, develop innovative models for geroscience research, and to advance novel uses for pre-existing technologies currently within the USC-Buck Nathan Schuck Center research cores and across existing USC-Buck facilities. We implement a voucher program that provides discounted or free access to cutting-edge technologies available either at USC or the Buck within the research cores. And third, as Sean was mentioning, we're going to be supporting research sabbaticals as COVID rules start easing up in aging and geroscience. So these research sabbaticals are designed to allow scientists, senior postdocs, or junior faculty from any academic institution in the US to spend a period of time directly interacting with Buck and/or USC faculty and research staff. It can be as short as a few weeks in order to learn the basics of important research models or new techniques such as Simon was showing you, or an extended research sabbatical to advance a novel project and foster new collaborative approaches. The goal is to foster a new generation of geroscientists comfortable working in interdisciplinary teams. These awards follow the format and expectations outlined for the P&F grants, except the awardees are embedded in a lab and our geroscience technology core at USC or the Buck for a period of 2 weeks up to 6 months. The trainees will attend weekly seminars and meet with speakers one-on-one, as well as at lunches and dinners at both campuses. The trainees will be asked to present their ideas and data at internal research seminars and group meetings on both campuses. Financial support from the Research Development Corps will be used for support travel and accommodation costs to supply research and reagent expenses associated with the project. And participants may propose to spend time at one or both institutions, depending upon how the project works and how collaborative it may be across the two institutions. So these are the applications that we've only had one round so far. These are the applications that were approved from round one. So there were 14 funded applications, and they're listed here. And the amounts of money range from about $7,500 to about $20,000, depending upon the needs of the project and the expenses associated with that project. As I say, none of these so far are sabbaticals because of the COVID restrictions, but as we start easing up, we hope to definitely be able to start funding more, funding these fellowships and sabbaticals. So how to apply, and Sean has already mentioned some of that. Contact core leaders to discuss feasibility of potential project And discussing the feasibility, as Sean said, we think is a really important step before you start making an actual proposal. And then to submit an official application through the Nathan Schock website. And here's the web address. Administrative questions, including budget, can be directed to Kira Harvath at the Buck. And there's Kira's contact information, email, and she can give you more information or directly interact with you on budgeting as the needs come up. That's my presentation. Thank you very much.
Thank you, Kelvin. So the next core is the Cellular Senescence and Beyond core, which is led by Judy Campisi and Birgit Schilling. And I think Birgit is going to be presenting.
Birgit, you're muted.
When I share, I cannot unmute, unfortunately. So I had to unshare, sorry. Judy, if you like to start, maybe?
Okay, I can start and then perhaps then Birgit can
Yes.
take over. So as many of you know, senescence has become an extremely important driver of multiple aging phenotypes and pathologies. We don't consider this core, though, to be totally focused on senescence. There are other cell fates that also can impact aging, and that includes things like apoptosis, as well as a new process that we've recently identified called cell competition. So our core will provide advice, but also standardized methods to really interrogate these cell fate decisions and ask whether or not they have an impact on your favorite aging phenotype or pathology. Thank you, Birgit. Next slide. So the first aim will be to advise on strategies, and for anyone interested in using this core, please contact either me or Birgit. It doesn't matter which one, because we will get back to you together, because we work very closely together, and advise you on whether the project is feasible, and also what we would best be able to do to help answer the question you're interested in. So the first will be to provide you with advice, and the second aim, if you can give the next slide, will be to also help you understand whether your favorite phenomenon is cell autonomous or cell non-autonomous. And to be honest, right now, most of the projects that we funded have been focused on cell non-autonomous effects, which means the biggest burden is on Birgit, because she does this amazing mass spectrometry, which helps you identify what proteins are being secreted by the cells of interest and how they might affect neighboring cells. So I think Birgit will now take over and give you an example of the workflows that she's developed, and also what's possible in the future.
Yes, oh, thank you, Judy. Yes. So yeah, as Judy mentioned, we do a lot of things together, and even if a pilot project decides they would like to do mass spectrometry, Judy's knowledge about senescence definitely comes in the interpretation of the results. So you'll definitely always work with both of us, which is really fun. I mean, so there are examples of things one could do. One could, for example, think about pilots where you induce senescence, and we have published this in a recent paper here by Nathan Basisti and Judy and me. And so you could look at how a certain cell type that you're particularly interested in undergoes senescence, and then how does the SASP look? How do exosomes that get secreted look? We actually published a pretty comprehensive protocol paper about this recently. And, you know, or you could think about, okay, how do these proteins in the SASP overlap? Or for example, one protein project that we have with Dan Weiner, he's actually looking at, you know, growing cells on different tensions, and then the effect of cellular senescence. So if you have creative ideas how you would like to investigate senescence and their secretory phenotypes or other things, or markers that Judy has available in her portfolio, we would be happy to work with you as part of these projects. And then really other things that you could also think about is, could you use some of these potentially as biomarkers, right? Would you be interested to look in some of these SASP markers in some biological fluids? We had recently published like the SASP atlas I mentioned earlier. And then we have a lot of really modern mass spectrometric instrumentation in our lab. And here this shows the Orbitrap Eclipse, very nice instrument. And we're doing very kind of highly quantitative workflows called data-independent acquisitions that should give you comprehensive acquisition, unbiased discovery, and accurate quantification. And then, of course, the knowledge that we have built in our labs over the last few years that we could compare with your results. So that's really, there's a lot of opportunities, I believe, if you wanted to work with me and Judy. And Judy just wanted to maybe briefly mention again some of the projects that we had for this year.
Yeah, so as Birgit mentioned, these are funded projects to look at the effects of mechanical tension. That is what the cells experience, both in a culture dish, but also in vivo, and how that might regulate what the cells secrete, and it's clear that it does. With Sean, also, again, mass spec profiling of the bacterial diets that are fed to C. elegans. And then finally with Bernice, also looking at how exogenous DNA or transposon-induced DNA can alter the phenotypes of fibroblasts as well as their secretome. So those are the three projects that are funded. This is how you apply. And again, please contact, it doesn't matter whether you contact me or Birgit, because we'll set up something where you'll talk with both of us.
Great. Great. Thank you.
Thank you.
Thank you, Judy and Birgit. And then so our last core presentation is the Genomic Translation Across Species core, which is headed up by M. Arpawong and Eileen Crimmins.
Hello. So I'm M. Arpawong. I'll be presenting for the both of us today. As Sean mentioned, we are the Genomic Translation Across Species core. And let's see here. So
I think you're not in presenter mode. Do you want to swap the slides?
Yeah.
Oh. Let's see.
Hey, this is Natalie. You can just click swap displays when you do that.
Yeah. Oh, can you see it now?
Yes,
The
so you just
slide?
go present from the current slide.
Mm-hmm.
But it doesn't matter. I mean, we can sort of see them, right? So rather than waste time, I think we can go with the way it's displayed now, no?
If you can see it?
Yeah.
And we'll go
Well,
with
we
that.
can. It's not in presenter mode, but we can see your slide. Yeah.
Okay. So we'll move forward with this for time. Okay, so the goal of the Genomic Translation Core is to advance translational research through collaboration. And so you can see here, most of you are doing research in your model organism and finding potential gene candidates that have effects on phenotypes or endophenotypes that are related to aging and lifespan. And then where we come in is here in stage 3, where after your primary findings, if there is a need to then validate some of your findings in human cohorts, that's where we come in. So we invoke natural studies of human aging. They're large-scale population-based studies with long follow-up follow-up times and repeated measures. And we include all of the geroscience hallmarks of aging. So we provide replication and race and ethnic diversity in the backgrounds. And what we do is we would conduct genetic association tests, transcriptomic tests, epigenomic analyses, bioinformatic annotation, and pathway analyses. And so that's shown here in box 3, where we fit in this cycle of research. And hopefully that would, these findings in the human samples would help to generate further research directions and hypotheses that could feed into informing more experimental studies and designs. So what we ask from potential project PIs for collaboration includes kind of a clear set of aims and hypotheses to interrogate, and which components you'd like to invoke that represent some of the phenotypes that you're interested in. And some of them that we have include data that come from blood-based biomarkers, including inflammatory and immune markers, for example, lipids, cytokines, glycated hemoglobin, red blood cells, and blood cell types. And then we have genetic data, epigenomic and transcriptomic data, telomeres, flow cytometry data, including T cells and B cells, monocytes, natural killer cells, physical health and metabolic parameters, such as BMI, blood pressure, and we have indicators of muscle functioning, disease status, cognitive and behavioral outcomes, and mortality and longevity outcomes. So here is a quick example of one project that we completed with investigator Via from the Curran lab. And we addressed a hypothesis regarding associations between a gene variant and aging-related muscle function decline in humans. And so what's shown here on the x-axis are multiple phenotypes that we looked at related to muscle functioning. And then on the x-axis are the p-values for the effect sizes to indicate the strength of association with particular genetic variants that are shown by the colored dots. So each color represents one specific genetic variant or a single nucleotide polymorphism. So this is an example of what we call a PHEWAS, a phenotype-wide association study with a gene. And so as shown, we're able to validate the relevance of some genetic variation within this gene region and natural processes of human aging. Just for example, we looked at a couple things like grip strength, decline, lift strength, walking ability, gait speed, physical mobility, and functional large muscle mass group ability. So this is how you can contact us.
Thank you very much, Em. So there's a couple questions in the Q&A and a few that were sent to me in the chat. So we'll go over those now. If you have other questions, please, you can start posting them and we will answer them. The first is this exclusive to faculty in the United States? And so the funding through the Nathan Schock Center is, but that said, the Buck Institute and the Davis School of Gerontology have a number of ongoing international partnerships. And so that shouldn't limit you from contacting us, having the Nathan Schock Center be the mechanism that gets you in touch with us. That's completely fine. You can send any of us core leaders, Gordon, myself, an email and we can assist you. That said, if you already have a partnership, if you are an international scholar and you have a partnership with someone in the United States, they can be the sponsor of a project. And again, the goal is to promote geroscience research. And so please contact us and we'll figure out a way to make it work. In the chat is the question that we get every single time, which is how much money can we get? And what is the difference? And the other question, which I'll answer together, is what is the difference between a voucher and a pilot? Okay, so it's really not as complicated as it might seem in the literature that we have out there. A voucher you should think about as USC or the Buck has a instrument or a technology that we want to use. How much does it cost to use it, right? And so the answer to that is that we provide it to you for free using the Nathan Schock Center as a mechanism to defray the cost for the staff that will run it, for reagents that are needed to do the analysis. You essentially send us the sample and we run it. And so, you know, these sort of vouchers are in the couple $1,000 range. And so if you have, that's usually what we give them for. Don't let the number be the thing that discourages you or the thing that you're gonna fixate on. Contact a core leader, contact Gordon and myself. We can get you in touch with the right person, and we'll let you know how much things cost. The goal is to make sure that the voucher or the pilot is sufficient so that it will cover all of the costs of that work. And we can figure that out for you. But vouchers are, again, you should think of them as an experiment, right? So we are doing an experiment for you. I think Gordon and I are most excited about the mini sabbatical sort of mechanism, because this is an opportunity for you to come to us and learn how to do it yourself, right? And so you can think of it as a voucher plus, right? It's the cost to do the experiment you want to do, but you coming to us and we can teach you how to do it hands-on. And those are going to be starting pretty soon. And then a pilot is a larger scale project. It's going to take more time. It's more than just running a sample, right? It's more that it's both project development. It's figuring out, you know, what sort of data analysis is going to need to be done. How much assistance do you need from project conception to project completion? And you know, and those can be in the low tens of $1,000s range. And so. Again, the most important thing that I can't stress enough is that you contact a core leader, you tell them the scope of what you want to do, they can tell you what's feasible and what's not, and then help assign a budget to make sure that your needs are met. So that actually answered, I think, 4 of the questions that were in the chat at the same time. Again, all of this information is on our website, and all of this is being recorded too, if you want to share it with your colleagues or your trainees. That said, we can wait a couple more minutes if people have questions. You want to raise your hand and ask one, or you want to type it into the chat or the Q&A. We can do that now.
Sean, while people are doing that, I just want to add a couple of things. One is that we're very much encouraged to collaborate between the Nathan Schock centers. And so you may have an idea that you're already looking at another Nathan Schock center and thinking about working with them. That doesn't preclude you working with us as well. And actually that would be really exciting to see projects that bridge between the centers. And maybe you do some experiments with us, and then you go into another center or vice versa. So look at all the Nathan Schock centers when you're thinking about this, and think about creative ways that you can build bridges between us. That would be really exciting. And the second thing is, you know, obviously one of the major goals is to bring new people into the aging field. It seems like a difficult goal. How do you get out there and find those people? But other centers have been able to do this over the years. And so please just help us by spreading the word and encouraging colleagues who even mention the word aging in passing to have a look at what we do.
All right, I don't see any additional questions. But again, look at our website, reach out to a core leader, reach out to Gordon or myself, and we're happy to get you in touch with the right people. And again, the money is there for you guys to use. So please contact us. Thank you for coming.